In the next few minutes, I'm going to tell you how we use nanopore sequencing to study disease-causing insertions. We work with a disease called X-linked Dystonia-Parkinsonism. That is an adult-onset neurodegenerative disorder, and it affects mostly individuals of the island of the Panay in the Philippines. It has been found that the disease causes neuronal cell loss in the striatum of the patients. But the aspect of the disease that I am very interested in is that recently it has been described that there is a polymorphic transposable element insertion inserted in the 32nd intron of the TAF1 gene. See, you can see here the schematics of the TAF1 locus in healthy individuals and then in XDP patients. TAF1 is a very important gene for proper transcriptional function, so you can imagine that its correct expression is very relevant. So the main question of this XDP field now is how does this transposon cause the disease? And that's what we are aiming to answer by using our neural progenitor cell model, where we have induced pluripotent stem cells from healthy individuals and XDP patients, and we converted them to these neural progenitor cell lines that we use in most of our analysis. So the first thing what we did is to do bulk RNA sequencing, and interestingly what we found is that in the intron of the TAF1 gene where this transposon is inserted, there is a retention of the intron only in XDP patients that you can see here with the tracks in green. And also this is highly statistically significant, this intron retention. So next we were wondering how is this transposon regulated, because we know that transposons are usually regulated by DNA methylation, but we don't know how these new polymorphic insertions that are not in the reference genome, how they are regulated. So that's where nanopore sequencing was very handy to us, because you can, as you all know, you can look at the methylation patterns. So then we did nanopore sequencing, and we looked at this XDP locus. As you can see here, as expected in the healthy individuals, there is no insertion. And then in XDP patients, marked with the orange field, you can see that this insertion is fully methylated. That is showed with the black circles. And then we asked the questions, now that we think that this methylation may protect this genomic region, we asked the question, what happens if you remove this DNA methylation? So by using CRISPR technology, we removed the DNA methylation in this cell type, and then what we found is that the intron retention increases greatly. So yes, indeed, the presence of this DNA methylation, it protects the genome from the effect of this insertion. And then we know that XDP is an adult-onset age-related neurodegenerative disorder, and we also know that DNA methylation can decrease with age. So now the next logical question is that how does this methylation look in the brain of the patients? And maybe this region is demethylated in the patients, and that's why they get the disease later on in their life. To do that, we again used nanopore sequencing, and this time we used post-mortem brain tissues from XDP patients. And here I just show you some preliminary results that we just start to dig in. And you can see here that, again, in the healthy individuals, as expected, there is no insertion. And in XDP patients, in the brain, this insertion has a dip in DNA methylation. So with that, I would like to conclude and just say that this XDP transposon causes intron retention of the TAF1 gene. This XDP transposon is covered in DNA methylation that decreases with age, showed by our preliminary data, and that the epigenetic status of this insertion is very important for the severity of the molecular phenotype of the disease. And it looks like restoration of DNA methylation could be a future therapeutic approach for these patients. And with that, I would like to thank for the Jacobson Lab and all our collaborators and our funding agencies, especially for the Collaborative Center for XDP here at Massachusetts General Hospital.